Okay, look around the room for just a second. Welcome to This Explainer. Today, we're going to map out exactly how that physical light bouncing off the world around you right now transforms into your conscious perception of reality. It's honestly a biological miracle, and it happens in fractions of a second every waking moment of your life. So get ready to look at your own vision in a whole new way.
Here's our roadmap for today. We'll start with the anatomy of the eye, then move on to capturing light and detail. After that, we'll look at what happens when vision goes wrong, trace the brain's visual pathway, explore making sense of sight, and wrap up with how we control eye movements. Let's dive in.
Section 1. Anatomy of the eye. Think of this as the hardware phase of our visual system. This is the biological camera taking in the physical light from our environment.
Let's track the actual path of light. Notice that highlighted area at the very back of the eye. That's the retina. Light enters, travels past the clear lens, moves all the way through that jelly-like vitreous humor in the center, and finally strikes that back wall. This extremely thin sheet of tissue, that's the true starting line for our biological relay race.
Moving to Section 2, capturing light and detail. Once light hits that retina, a microscopic cellular showdown takes place.
Inside the retina, we have these specialized cells called photoreceptors, and they basically come in two main teams. First up, the rods. Think of them as your night watchmen. They hang out in the periphery, they're highly sensitive to low lights, and they only see in black and white. You know when you're stumbling through your house in the dark? Yep, that's your rods at work. Then we've got the cones. These are your detail-oriented artists. They're packed really tightly into the central part of your retina, an area called the fovea. When you're admiring a colorful painting, that's your cones taking over, giving you sharp, daytime, high-definition color vision.
Now, once those photoreceptors capture the light, the signal doesn't just leave the eye right away. They hand the baton to bipolar cells, which act as the very first true neurons in this pathway. From there, the signal goes to specialized ganglion cells. We've got parasol ganglion cells, which are built to detect gross movement. And on the flip side, we have midget ganglion cells, which are perfect for capturing fine visual detail and color. What this means is absolutely wild. Your eye is already pre-processing motion and detail before the signal even reaches your brain.
That brings us to Section 3, when vision goes wrong. To really understand this incredible hardware, we need to see what happens when specific parts of it break down.
The crucial point here is understanding conditions like age-related macular degeneration, or AMD. AMD involves damage to the macula. Now, the macula is that oval region right in the center of the retina surrounding the fovea. Remember those detail-oriented cone artists we just talked about? Well, because this area is absolutely packed with them, damage here means we lose our high-acuity central vision.
Imagine trying to read a book or recognize a loved one's face. To a patient with AMD, it looks just like this. The periphery is fine, but the center becomes a really frustrating blur. The damage is located centrally in the macula, literally right where you'd naturally look to focus on something directly in front of you.
Now contrast that with glaucoma, which behaves very differently. It's caused by increased intraocular pressure in the eye. That pressure pushes outward, and it causes damage to the outermost optic nerve fibers first. Because those outermost fibers are the ones carrying data from the peripheral edges of the retina, a patient with glaucoma loses their peripheral vision, which results in these severe obstructing black patches around the outer edges of their sight.
Let's move into Section 4, the brain's visual pathway. Here, we're going to follow our visual signal out of the eye entirely, tracking it deep into the central nervous system.
As the optic nerve leaves the eye, we encounter this fascinating neurological plot twist. The nerve fibers from the nasal side of your retina, so the side closest to your nose, actually cross over to the opposite side of the brain at a junction called the optic chiasm. Meanwhile, the fibers on the temporal side, near your temples, they just stay on the same side. Because of this elegant little crossover, the left side of your brain ends up processing the right side of your visual world, and the right side of your brain processes the left side. It's a biological quirk scientists believe evolved to help us with depth perception. So let's map out how this builds into the complete retinogeniculate pathway. Think of this as the main highway for our conscious vision. The signal starts at the retina, shoots down the optic nerve, navigates that optic chiasm we just mentioned, travels down the optic tract, makes a critical pit stop at the LGN and the thalamus, and finally terminates at the back of the brain in the primary visual cortex, otherwise known as V1. When that signal hits the LGN and the thalamus, it doesn't just get jumbled together into a mess. It is highly organized.
Input from those parasol ganglion cells we mentioned earlier, the ones tracking gross movement, goes exclusively to the magnocellular layers. Meanwhile, the input from the midget ganglion cells, the ones tracking fine detail and color, routes straight to the parvocellular layers. Your brain literally keeps the signals from motion and detail strictly separated in entirely different physical layers. How cool is that?
All right, Section 5, Making Sense of Sight. The signal has reached the back of the brain, and now it's time for the software to decode these electrical signals into a cohesive, meaningful reality.
From the primary visual cortex, the data splits into two massive information streams. First, the parietal or dorsal stream goes upward. This is your where pathway, constantly calculating spatial relationships and tracking how things are moving. Second, the temporal or ventral stream goes downward. This is the what pathway, and it analyzes detailed form and color to actually identify what an object is. This leads us to a truly mind-bending question. If these visual streams handle object identification, what happens if your brain loses its specific facial recognition software?
Well, the answer is a condition called prosopagnosia. Deep in the right inferior temporal lobe, we have populations of recognition neurons that are finely tuned specifically to recognize faces. Imagine looking at your best friend. You can see their nose, their eyes, and their mouth perfectly sharply, but your brain completely fails to assemble those isolated features into a face you actually recognize. That's prosopagnosia. It's a powerful reminder that merely seeing something and actually recognizing it are two totally different brain functions.
Finally, we reach our last section, controlling eye movements. Because our visual processing is incredibly advanced, sure, but it's utterly useless if we can't aim our eyes at what we want to see.
Aiming our eyes requires a massive, complex internal communication highway. First, the visual cortex spots a target and decides to look at it. It then sends a signal down to gaze centers in the brainstem, which command horizontal movement. But to move both eyes together, a special bridge coordinates multiple nerves instantly, ensuring that the muscles of both your left and right eye pull in perfect unison. All of that computation, just a glance to your left?
This amazing system allows for saccadic eye movements. Saccades are incredibly rapid, completely subconscious eye movements that we make constantly. In fact, your brain is aggressively darting your eyes around your environment right now as you listen to this, reflexively ensuring that the most important details land precisely on that tiny, cone-packed fovea at the center of your retina.
Every single time you shift your gaze, your visual system subconsciously analyzes a target, calculates exact 3D spatial coordinates, routes the data through brainstem gaze centers, and fires precise cranial nerves to move physical muscles. And all of this happens in a fraction of a second, with every single glance you take. It's truly staggering. But it leaves us with a pretty fascinating question. If our eyes and brain do so much editing, filtering, and pre-processing before a visual signal even registers in our consciousness, how much of what you see is objective reality, and how much is just your brain's interpretation? Thanks for joining me on this explainer. I'll see you next time.